continuing with the class human health and diseases. In last class, we have learned about the definition of health. We have learned about the causative agents of diseases. Right? So mainly when we are talking about the human diseases, we have to remember about the causative agent, like the helminths or protozoans, right? Or viruses or bacteria that causes a particular disease. And we have to learn about the mode of transmission of the disease, the incubation period of the pathogen, the symptoms of the disease, and the particular treatment of the disease. So today we will learn about the bacterial diseases. That means the diseases which are caused by bacteria. In the last class we have learned about the viral diseases. So today we learn about the diseases which are caused by the bacteria. And under this one, we mainly learn about the two diseases, typhoid and pneumonia. So typhoid and pneumonia are mainly the bacterial disease which we are going to discuss today. So first of all, typhoid, alright, also called enteric fever. Now pathogen, that will be causative agent that causes typhoid. So therefore, the causative agent of typhoid is mainly a rod-shaped bacteria. The shape of the bacteria will be like a rod shape here, and therefore the scientific name of this rod-shaped bacteria is Salmonella. Can you remember? Salmonella typhi. Okay, so therefore the scientific name of the bacteria that causes typhoid is Salmonella typhi. And maybe it infects the intestine here. So this Salmonella typhi is a rod-shaped bacteria, alright, which causes acute infection of the intestine. So to remember, causative agent. Now mode of transmission, how are these pathogen or bacteria transmitted from an infected individual to a healthy individual? So when we look into the mode of transmission, this particular pathogen is mainly transmitted by the help of a carrier. Now try to recall the difference between a vector and a carrier. Clear? So this particular bacteria is mainly transported from one place to another place by the help of carrier. And one example of carrier is house flies. Clear? So whenever the house flies will carry the bacteria from the fecal material of an infected person to the water body or to the food that we eat. So at that particular time, the food that we are taking, which are contaminated by this bacteria, then will also become infected by this bacteria and will become typhoid. So look over here. So disease spreads through the food and contaminated water. Now what do you mean by contaminated water? Those which contain the bacteria Salmonella typhi. Here. Now, mainly those who are infected with the typhoid, this particular bacteria will be excreted through their fecal material, through their feces. So housefly is the carrier. They will carry the pathogen from the fecal material or from excreta to the food, to the water, or any other eatables that we used to eat. Okay, so pathogen which are present in those eatables, like in food, water, milk, or any other eatables, will enter our body through the mouth and will reach the intestine and causes lesions in the intestinal wall. Here, this area of bacteria has entered our body that infected our intestine. So therefore now, the incubation period, that is the time period between the entry of germs into our body with the appearance of first symptom, which is called incubation period. So the incubation period of Salmonella typhi varies from 1 to 3 weeks. So 1 to 3 weeks is the incubation period of Salmonella typhi. Now what are the symptoms of typhoid? So individuals who will suffer from typhoid will have high fever in the first and second week, and that will gradually decline. Alright? During the fourth or third and the fourth week, there will be severe body pain. Okay, there will be weakness, there will be loss of appetite, there will be constipation and stomach pain. So these are the symptoms of an individual who are infected by Salmonella typhi. Now, liver and the spleen becomes enlarged. In severe cases, intestinal perforation. Now, what do you mean by perforation? Perforation means the formation of pores. Clear? So, intestinal perforation means the pores will be formed in the wall of the intestine. Clear? So, intestinal perforation and death may occur. So, the pores may occur on the wall of the intestine, and eventually that individuals may even die. Typhoid fever can be confirmed by which test? By the viral test. Can you remember? Typhoid can mainly be diagnosed right at the earliest by the help of the viral test. W-I-D-A-L. Viral test. So viral test is the test which is mainly performed to confirm the disease typhoid, which is mainly caused by Salmonella typhi. So you can look over here in the diagram. This particular diagram which you are seeing over here is mainly the rod-shaped bacteria Salmonella typhi, which causes typhoid. And therefore, viral test is mainly the agglutination test of antigen and antibody, which will help in the early diagnosis or the confirmation of typhoid. Clear? So, how can we prevent the transmission of this Salmonella typhi, or what are the preventive measures of this disease? So, first one, we must maintain proper community sanitation. Cleanliness, hygienic condition must be there. Clean and chlorinated water supply system, that means drinking water, must be free from contamination of those bacteria, and personal cleanliness is most important. Prevention of food contamination by the flies and dust. Cooks and food handlers in eating establishments should be pressed upon to wash their hands with soap and water before handling. That means, let's say for example, if any one of an individual who is dealing with food handling, or suppose let's say here is a cook, and suppose that individual is suffering from typhoid, or let's say here she is not showing the symptom of the disease typhoid, but they are the carrier. In last we have mentioned that an individual may be infected by the pathogens, but he or she cannot show the symptom of the disease. In that case, they will be the carrier of the pathogens. Over here, let's say, if that individual who is a cook, and suppose that individual is a carrier of Salmonella typhi, then in that case, those bacteria will be transmitted from that cook to the other individuals. That means he is the carrier and he is transmitting the disease typhoid. Clear? So in that case, those who are dealing with cooking purposes or handling vegetables or with food handling, they have to wash their hands frequently with soaps. Clear? So that the food or any other eatables will not be contaminated by the Salmonella typhi. Now, treatment we can say, typhoid vaccines, it provides immunity for about 3 years. Rest, care about food and proper nursing essential for some days in order to prevent the relapse. But most important thing, we have to give the typhoid vaccine. Okay, the most important one. Then, control. There are certain drugs which can be given to those individuals who are suffering from typhoid. So the name of those drugs are ampicillin and chloromycetin. So kindly remember the name of the drugs which are given to those individuals suffering from typhoid. The name of the drugs are ampicillin and chloromycetin. So these are the drugs which can be administered or which can be given to those infected individuals suffering from typhoid. Clear? So remember the name of the carcinogenic agent that causes cancer, incubation period of the bacteria, symptoms, then preventive measures, then the name of the drugs which can be given. Clear? Now, next one, we talk about pneumonia. The second disease is caused by the bacteria. So pneumonia is also caused by a bacteria. The name of the bacteria that causes pneumonia are mainly Streptococcus pneumoniae. Remember, Streptococcus pneumoniae and Haemophilus influenzae. These are the name of the two bacteria that causes pneumonia. So first one is Streptococcus pneumoniae and second one is Haemophilus influenzae. So this particular bacteria, they will infect the lung alveoli. Now you know what I mean by alveoli. The answer, right? There will be exchange of gases. So mainly they will infect the lung alveoli. Clear? So what is that? Due to the infection by this bacteria, the alveoli of the lungs will be filled with fluid. Clear? So due to the infection by this bacteria, the alveoli of the lungs will be filled with the fluid, leading to severe problem in respiration. So there will be a respiration problem in those individuals suffering from pneumonia. Clear? The mode of transmission of this disease. Before that, we will just see the diagram of these two types of bacteria. You can see over here. So this one is Streptococcus pneumoniae and this one is Haemophilus influenzae. Clear? The first diagram you can see over here is of Streptococcus pneumoniae and this particular diagram is of Haemophilus influenzae, which causes pneumonia, which will mainly infect the lung alveoli. Now, let's talk about the mode of transmission of this bacteria. So, a healthy person gets infected by this bacteria in what particular conditions? Number one, when a healthy individual inhales the droplet released by the infected person. So, whenever an infected person will cough or sneeze, droplets will be released in the air. That droplets will contain the bacteria. So when healthy individuals will inhale those droplets, then he or she will be infected by this bacteria. Clear? Let's look at the conditions. By sharing drinking glasses, utensils, food or water with an infected person. So by sharing of articles or things between the infected persons, this particular bacteria can be transmitted from an infected individual to a healthy individual. Clear? Next condition, by the sputum of the patient. Alright, so even from the sputum, this bacteria can be transmitted. So what are the symptoms of the disease? What are the symptoms of pneumonia? So there will be fever, there will be chill, there will be cough, headache, chest pain. In several cases, lips and fingers, nails may turn gray, tuberous in color. Okay, there will be bloody and rusty sputum. So these are the symptoms of pneumonia. There will be abdominal distensions or discomfort. So depending upon the location of infection, kindly know this one. Depending upon the location of infection, there are two types of pneumonia. Alright, so we mainly mentioned that this bacteria will go and infect the lung alveoli. So depending on the location of infections, where these pathogens will go and infect. So mainly there are two types of diseases you can see. First one is bronchopneumonia. Remember, bronchopneumonia, bronchitis. Readable is written, bronchopneumonia means bronchitis. So bronchopneumonia will inflate the air sacs. And number two is lobar pneumonia. Alright, lobar pneumonia, L-O-B-A-R, lobar pneumonia, which will infect the pulmonary lobe. Clear? We have got two types of diseases or pneumonia depending upon the location of infection. So we have got bronchopneumonia, which will inflate the air sac, and number two is lobar pneumonia, which will entirely infect the pulmonary lobe. Now, incubation period of these two types of bacteria is mainly 1 to 3 days. A very short time is there, just take 1 to 3 days to show the symptom of the disease. So incubation period of Streptococcus and Haemophilus is very, very short. It just takes 1 to 3 days. Okay. Treatment by the help of antibiotics, and what are the name of the antibiotics that can be given? Is erythromycin and tetracycline. So these are the name of the drugs which can be given to those individuals who are suffering from pneumonia. So the name of the antibiotics are erythromycin and tetracycline. Clear? So here I mentioned that the name of the bacteria, you can see over here, the diagrams. So this one is Streptococcus pneumoniae and this is Haemophilus influenzae that causes pneumonia. Clear? So can I do the figure also. Now, next disease, we will talk about the protozoa. That means the diseases which are caused by protozoa. Yes, so over here, we will go two diseases, amoebiasis, or we can say amoebic dysentery, and number 2 is malaria. So these are the two types of diseases which are caused by protozoa, or simply called it protozoan diseases. So today we will only discuss about amoebiasis. In the next class, we will go in detail about malaria, where we will talk about the life cycle of Plasmodium also. Clear? So today we will just talk about amoebiasis. So let us see about amoebiasis or amoebic dysentery, which is caused by the protozoan Entamoeba histolytica. Clear? So Entamoeba histolytica is the name of the protozoa which causes amoebiasis or amoebic dysentery. So they are mainly the parasites which are completely dependent on the host for their nutrition, or we can say they are completely dependent to complete the life cycles of the body of the host. Without the host, they cannot survive. So Entamoeba histolytica is the name of the protozoa that causes amoebic dysentery or amoebiasis. So this Entamoeba histolytica occurs in two forms. Kindly note it down. It occurs in two forms. The first form is called the trophozoite form. Trophozoite. The first form is called the trophozoite form or the Magna form. Magna form. Okay, so the first form is called Magna or trophozoite form, and the second form is called the Minuta or pre-cystic form. Clear? So this Entamoeba histolytica occurs in two forms. First one is called Magna or trophozoite form, and the second one is called Minuta or pre-cystic form. Clear? So this particular trophozoite form or the Magna form is highly pathogenic to men. Okay, this is pathogenic to men. It is a feeding form and it is pathogenic to men. It causes amoebiasis in human beings, and therefore the Minuta or the pre-cystic form is the non-feeding form and is non-pathogenic. Clear? So between the two, trophozoite form and the pre-cystic form, trophozoite form is the feeding and pathogenic one, whereas Minuta or pre-cystic form is the non-feeding form or non-pathogenic form. So you can see the structure over here. Now we will look in details about the structure of Entamoeba histolytica. So here we can see the structure. This one you can see over here is the Entamoeba histolytica trophozoite forms. Okay, so you can see over here the structures in detail. Alright, so all the structures are Entamoeba histolytica, which are infected with red blood cells. So this one is the trophozoite form. So here you can see clearly the structures. This is the form of Entamoeba histolytica, which is infecting the human being that causes amoebiasis or amoebic dysentery. Yeah. Now we learn about the infections, mode of transmission, symptoms, prevention, and treatment. So infections. So it will mainly infect the upper part of the colon and the lower part of the small intestine. For example, like in case of pneumonia, we have learned that it will infect the lungs alveoli, right? So here, in case of amoebiasis, the protozoal intramoeba will infect the upper part of the colon and the lower part of the small intestine. Clear? So it will invade the intestinal wall, it will invade the intestine, it will destroy the mucosa and will feed upon the red blood cells. So this particular intramoeba can be seen under the microscope in the red blood cells. Okay. Now, it causes ulcers or abscess. Abscess means it will create the cavity or formation of cavity. Okay, so it will create ulcers, it causes ulcers, or it will form the cavity or the abscess in the intestinal wall. The ulcer will burst, releasing blood and mucus, which comes out with stool and causes aerobic dysentery. It will lead to the formation of the ulcer, it will lead to the formation of the cavity or the abscess, alright? The ulcer will burst, it will rupture, and therefore the mucus will come out with these stools, leading to aerobic dysentery. Now, how does these protozoans will be transmitted from an infected individual to a healthy individual? So it is mainly transmitted through the contaminated food and water. Yes, so whenever the food and the water will be contaminated by these protozoans, and if one of the healthy individuals will take up those contaminated food and water, then that individual will be infected by this particular disease. Now, symptoms: blood and mucus in the stool, abdominal pain will be there, nausea, flatulence, bowel irritability, fatigue and headache will be there. Preventive measures: washing of hands frequently or taking meals, protection of food and water from flies and cockroaches. That means we have to cover the food, alright, so that it will not be contaminated by those protozoans. Proper sanitation needs to maintain. Raw vegetables should be soaked in potassium permanently before usage. Adequate cooking of food to destroy the sick. So food or vegetables must be properly cooked so that the cystic form of those pathogens or those protozoa can be destroyed or can be killed. So avoid taking raw vegetables. Treatment: antibiotics can be given for treatment of amoebiasis. So the name of the antibiotics which can be given are fumagillin, terramycin, aromycin. These are the name of the antibiotics which can be given for the treatment of amoebiasis or amoebic dysentery. Clear? So for eradication of intestinal infection, so remove the infection of the intestines, certain arsenic compounds like carbazole, carbazole or biofilm are effective. Yeah. So kindly go through the diseases that are caused by bacteria and that are caused by protozoa. So on bacteria we have learned about pneumonia, we have learned about typhoid. So kindly remember that. Pneumonia conjugate agent, symptoms, preventive measures and treatment, and with mode of transmission. And protozoans we will simply have learned about amoebic dysentery caused by Pneumonia sonatica. In the next class, we will learn about the second disease caused by protozoan, that is malaria. It is mainly caused by plasmodium. So there are different species of plasmodium which causes malaria. Okay, so we will go in detail in the next class about malaria, and we also see the life cycles of plasmodium. So this much for today. See you in the next class. Thank you.